So next, I'm going to just walk through the display of a data set. So this is how all of that information then gets rendered. And I know that on the far right-hand side, hopefully that's right for you, it's right for me, that's a small thumbnail, but it's really, that's just kind of like our roadmap. I'm gonna just blow up different parts of that as we go through, because that's clearly too small to see there. But we'll just get right into that. So at the top, what we have is a summary of the data set. This begins with persistent identifiers, so, or really a title, and then persistent identifiers. Every data set in the system has a persistent record ID, and along with it, for data sets, we integrate with the data site service to generate digital object identifiers. So this is the exact same identifier scheme and mechanism that journal publishers would use. So we think of data as a first-class resource, and as such, it needs an archive-grade identifier. So our preferred way of referring to data is with the digital object identifiers. Everything throughout the system has a record ID, and that is still a unique identifier throughout the entire database. And when I say everything, I mean things that maybe don't have the DOIs, like the individual experiments and the biosample records and the replicates, and every unit of data has this internal record identifier, which is kind of our new scheme of an accession number in FaceBase. And then, again, to emphasize the citability of FaceBase data, we have all the attribution details. So a link there to the project, and then individual contributors. So you can think of these as the equivalent of the authors of a paper. And these go into the citation information for a particular data set. And then next are some top-level tags or keywords associated with the data set, and these are categorized into different classes like experiment type, species, stage that are shown here, and there are several others as well. And now going back to what Chris mentioned in one of her last slides there about the FAIR data. So the I in FAIR is interoperable, and to be interoperable, we need to use terminology that is understood outside of just FaceBase, outside of just your lab. And to do that, we source in terms that have been provided from the biomedical research community, and for those doing more the bioinformatics and library of medicine and things like that. So these terms come from external vocabulary sources, and so they have well-defined identifiers and well-defined meanings. And so we really emphasize using those wherever possible. If it's not possible, then inside of FaceBase, we can mint our own accession and identifier. So at least we know, even if it's FaceBase locally coded, we have a consistent way of referring to it. But we prefer, where possible, always pulling sources of terminology from external standards. The next part of a dataset page is that visualization component. So these are generally available right after the initial summary and kind of give you a way to see some idea of what's in the data set. These can range from thumbnails to, here we see a genome browser embedded window in the page. Additional types of information or visualization we have vary based on the type of data set. The top two are available today. The bottom two are in the pipeline. So other types of data sets will have, say, volume data, CT, micro CT type of data. And for that, we can apply an orthogonal slice viewer to it. And so you can kind of rotate it around, zoom in or slice through the image along all three axes. In the upper right-hand part of this, the body of the slide here, we have the surface models. So what Chris showed you there in terms of our image navigation is also a tool that's available on all data sets. And so we don't have a lot of this data yet. We are encouraging future submissions to really include these if possible, because the nice thing about them, at least for hard tissue kind of imaging, especially, is that these are generally available right when somebody is working with the imaging data, the reconstructed images, and they can generally save out a surface model. And they're a bit smaller, so when someone is viewing them, it's a little easier quicker to load them up and rotate around and view them. So this is another general capability. And then, as I mentioned, the bottom two are in the pipeline. These are technical capabilities that we have, and there's data for these that, in particular for the histology data that is in the pipeline that we're expecting to receive in the next year about. So we expect to see more of this online. Of course, we're getting single cell data, and we look forward to being able to provide more of that visualization online as well. So technically, these capabilities are ready or about ready, and we are expecting to have data for at least some of them in the pipeline soon. Okay, and then moving to the next part, then you really get into the detail of that data set, because what we saw before at the top was just a summary. Now we really get into the exact specifics, and by the time we get down here, we know that we're interested in really drilling down and knowing more about it. And say, rather than just having a zip file dropped on us with some information, we're able to have like all of detail about the metadata, about the experiments. So each row here represents one particular data point for this data set. And we'll go into more of this in the live demo part, but I'll just mention that it comes towards the bottom. You see both those experiments and biosamples, as well as each of the classes of data files that exist and were submitted for that data set. Data sets can have a lot of data in them, can have a lot of data files. They can have a lot of that metadata, and you might like to be able to download those in bulk. Alejandro is going to show a demonstration of doing that, but I'll just point out that at the top of the page is this export menu, and in it there's a bag, and it's called the bag. It's a standard that came out of the library community, and we've adopted it in the biomedical, within the biomedical domain, and came out of some other NIH-related resources that have worked on this. And it's actually a format that is very good and tailored for large data files. So what you do is, say, okay, we're looking at an RNA-seq and ChIP-seq data set here. The data files are in the tens or maybe more of gigabytes. So you probably wouldn't want to download that in one giant zip file in a browser. But what you can do with this bag format is that you can download just the metadata and all of the identifiers for the individual files, along with their what's called a file checksum, and that's essentially a cryptographic signature, if you will, for the data that tells you exactly, it's used for integrity checks. It tells you exactly, how to verify that you got the data without any corruption in it. So you don't need to know the details of it, but that's what our bag format comes with. And basically our tooling that Alejandro should be able to show actually downloads and then verifies those checksums as it goes. And then lastly here, before getting to the demonstration part, I want to just revisit the topic of the data citation. So on every data set page, there is a sharing link, and I'll show this on a live page. But there is a sharing link that has both the highlight of the actionable identifiers, the persistent identifiers for the data set, as well as a little explanation here towards the bottom middle here under the data citation section, which show you a data citation reference to this data set should look like per some of the guidelines. Of course, you're probably going to follow whatever the formatting guidelines for your manuscript would be, but this is kind of this follows the general guidelines. And I believe this is out of the Nature Publishing Group as well. but others follow something similar to this as well. And then at the bottom of this window is this download data citation, and it has this name called BibTeX. And that is just a structured format that gets you all of the details, like all of the contributors who go in the author column and so forth. And you can download that file, .bib file, and most reference managers like EndNote and other will accept that. So you can basically import it right into your reference management software and then use that as you're working on a manuscript.